Hi and welcome to the Bio 181 Tutoring Center videos. Today we're going to be talking about genetics and the process of meiosis. This is your first lecture for Bio 181. Everything's already drawn out. It's kind of, there's a lot of information. It looks confusing to start with, but if you just follow this cursor, I'll talk you through everything and it will make sense, hopefully. If you get lost, of course, pause the video, rewind, watch it again, watch as many times as you need. Take lots of notes as well as you're watching this. I hope that this video helps. So as you remember from Bio 180, we have the cell cycle. You talked about the interphase. G1 is the replication of the cytoplasm, S phase, replication of the DNA, and the G2 phase is the final checks where we check the DNA, and then we move on to mitosis. Well, in this case, we're going to talk about meiosis, some of the differences. Mitosis is the process by which we replicate our somatic cells. Somatic cells are just regular cells, and mitosis just clones the cell. Meiosis, however, is a process by which we create gametes, so our gametic cells, and these are reproductive cells, so sperm and egg. It's a process by which we go from one cell to 4 new cells with half the amount of genetic information. So we go from 2n to n. Now before we go into that and talk about meiosis, we need to understand chromosomes. So we have lots of chromosomes, but scroll down. We have one from our father and one from our mother, known as the paternal chromosome and the maternal chromosome respectively. These are called homologous pairs. In humans, we have 23 pairs, or 23 homologous pairs, which means we have 46 chromosomes in total. This is one chromosome, this is another chromosome. The two together form a homologous pair, otherwise known as homologs. We have 22 pairs of autosomal chromosomes, and then we have one pair of sex chromosomes, so we have 2 sex chromosomes. The autosomal ones just code for everything basically in our bodies and all its functions, but then the sex chromosomes determine our gender, and they have a few specific functions as well. And so females have an XX chromosome and males have an XY chromosome. Now after S phase, the chromosomes are replicated chromosomes, and this is a replicated chromosome. Still one chromosome, but it's replicated. A lot of people get this confused and see this replicated chromosome and think it's 2 chromosomes likewise. But no, this is just the paternal and the maternal chromosome replicated. And so we have what's called sister chromatids. The 2 parts are called sister chromatids. This dense part in the middle, tightly compact DNA with cohesive proteins, and this kind of useless junk DNA is just repeating information. This is called the centromere and is important for cell division, for the separation of the chromosomes, and we'll talk about that in a minute. But just remember this is still one chromosome. Meiosis, as I said, is the process by which we go from diploid, or 2n, to n, or haploid. So we have one diploid cell and we turn it into 4 haploid cells, where n is the number of homologous pairs. So we go from 23 homologous pairs, or 46 chromosomes in total, to 1 times n, which is, we just go to 23. So we go from 46 chromosomes to 23 chromosomes in each of these cells. This could be sperm or egg. Remember, humans, 2 times 23, we have 46 chromosomes in total. It's an important number to remember. Now we understand that, we can talk about meiosis. Meiosis begins with prophase 1. Prophase, prometaphase, metaphase, these are words that you will recognize, and the processes are somewhat similar to mitosis, but there are some differences. In prophase 1, the homologous pairs line up. This green lattice structure here in the middle is the synaptonemal complex. These proteins hold it together so that genetic recombination can occur right here. And what that means, these two, the sister chromatids that interact and share DNA are called recombinant sister chromatids. The 2 that don't interact are called non-recombinant sister chromatids. And after this process is over, it looks like this. See the pink and the blue part have switched, so these 2 recombinant sister chromatids have literally shared genetic information. Where they remain attached after the synaptonemal complex is broken down, this is called the chiasmata. and the whole thing is called a tetrad right here. This is the number one source of genetic variation in humans and many organisms, and something to remember that. And then something else that happens in prophase 1 is the nuclear envelope begins to break down. The blue bit here is the nuclear envelope breaking apart, and then our spindle apparatus begins to form. In prometaphase 1, the spindle apparatus is fully formed and the nuclear envelope is dissolved. As you can see, it's gone, and these microtubules attach, these green bits of the microtubules, and they've attached to the centromeres, that densely compact region in the middle of the chromosome, and they're ready to pull the things apart basically. And then we go on to metaphase 1. This is the second source of genetic variation. It's known as independent assortment. The homologs line up along the metaphase plate. Metaphase plate is the middle of the cell, and the chromosomes line up together in pairs along this part, and it's completely random. That's what independent assortment means, is they line up randomly. So the pink and the blue one, the maternal and paternal chromosome, can line up on either side. It doesn't matter. It's completely random. And so the number of ways the homologs can line up is 2 to the power of n, where n is corresponding with the number of homologous pairs. And so 2 to the power of n tells you how many possible gametes there are. This is an important piece of information to remember. An example is if an organism has 8 homologous pairs, then 2 to the power of 8 is 256. There are 256 possible gametes this organism can have, or 256 ways that these chromosomes, these homologous pairs, can line up. In humans, we have 23 homologous pairs. 2 to the power of 23 is 8.4 million. So from every round of meiosis, we can produce 8.4 million different gametes, and that's not even including the events in prophase 1. So as you can see, humans have very high levels of genetic variation. Then we have anaphase 1, and this is simple. The spindle retracts and the homologs separate. So the homologous pair, both homologous pairs have been separated. Still 4 chromosomes. Then telophase 1 and cytokinesis. The cleavage furrow is constructed as actin filaments retract. They retract and divide the cell in half, and we're left with 2 haploid cells. We went from 4 chromosomes to 2 chromosomes in each. This is a simple bio 180. Remember the cleavage furrow. I hope you remember this. I'm not going to go into too much detail. And then just be aware that only in some species do the chromosomes decondense and the nuclear envelope reforms. As you can see here, I started drawing the nuclear envelope again. Then we move on to meiosis 2. We have prophase 2, where it's pretty much the same as mitosis. If the nuclear envelope reformed and the chromosomes decondense, then the chromosomes come back together, they recondense, and the nuclear envelope begins to deform again, break apart, and then the spindle apparatus here begins to form again. The microtubules are constructed towards the centromeres of the chromosomes. Then we go on to prometaphase 2. It's pretty much the same. This is simple, just the envelope's gone and the microtubules have attached, the spindle apparatus is completely formed. Metaphase 2, the sister chromatids, not the homologous pairs, but the sister chromatids have lined up. This is similar to mitosis, and so is prophase 2, similar to mitosis, that there's no crossing over occurring. And once this has happened, then anaphase occurs and the sister chromatids separate by the retraction of the mitotic spindle again. This is very, very similar. And then we have, this is very similar to the process of mitosis. Lastly, telophase 2 and cytokinesis. There we go. Telophase 2 and cytokinesis. The cell divides again with the cleavage furrow. The nuclear envelope begins to reform around the chromosomes that have been separated. And obviously I just followed one cell. There's another cell doing this. So at the end, we're left with 4 haploid gametes. 1, 2, 3, 4 cells with 2 chromosomes in each one, instead of what we started off with. We started off with one cell that had 4 chromosomes. So we've gone from 2n, 1 cell, to 4 cells of just n. Then finally, the last source of genetic variation is random fertilization. Basically, an egg cell from the mother and a sperm cell from the father, completely random, can come together, fertilize and form a zygote, form a baby, and it's completely random. So that's again another source of genetic variation. Let's recap. Crash course, let's just run through this quickly. Prophase 1, genetic recombination, the formation of the tetrad and the chiasmata. The nuclear envelope breaks down, spindle apparatus begins to form. Remember, the crossing over occurs between recombinant sister chromatids, and this is the number one source of genetic variation. Metaphase 1, the nuclear envelope is dissolved, and the microtubules attach. We go to metaphase, and they line up. Second source of genetic variation is independent assortment. They line up in the middle of the cell. Remember, it's 2 to the power of n is the number of possible gametes, where n is the number of homologous pairs. Anaphase 1, the spindle retracts and the homologous pairs separate. And then telophase 1 in cytokinesis, the cell divides and we're left with 2 haploid cells. Prophase 2, same as mitosis, there's no crossing over, and the nuclear envelope is beginning to deform if it reformed if it reconstructed here, and the chromosomes recondense if necessary. They recondense sometimes. Microtubules begin to form. Metaphase 2, the envelope's gone if it was reformed. The spindle apparatus is fully constructed. This is the shortest and not too important, but remember it at least. Metaphase 2, the sister chromatids line up, not the homologous pairs. Anaphase 2, sister chromatids separate, and then telophase 2 in cytokinesis, the cells divide again. We're left with 4 haploid gametes. Remember, meiosis 2 is very similar to mitosis. Now we've recapped. Alright, so I've got some practice questions here for you. Let's go over these. There we go. Alright, so if a cell contains 12 chromosomes, how many ways can the homologous pairs line up? Now it's important when you're reading a question to read it carefully and highlight the important information. So if a cell contains, and as you see here, I've highlighted 12 chromosomes and the homologous pairs lining up. So if you have 12 chromosomes, how many ways can they line up? Pause the video, figure it out, see what you get, and then I'll give you the answer right now, but pause the video now. So how we solve this is we remember that if there's 12 chromosomes, that means there's 6 homologous pairs, and 2 to the power of n is equal to the number of ways they can line up, and n is the number of homologous pairs. So we have 2 to the power of 6 homologous pairs. If there's 12 chromosomes, there are 12 chromosomes here, then that means there's 6 homologous pairs, so 2 to the power of 6 is the answer to this question. 2 to the power of 6 number of ways that the homologous pairs can line up along the metaphase plate in metaphase. And therefore, there are 2 to the power of 6 number of gametes possible. This next question, which of the following meiosis events are not the same in mitosis? Look at it for a moment. There's all these different options, A through H. Figure out, choose which one it is. Pause the video now, figure it out, and I'll give you the answer. So the answer is H. C, D, and E. In C, we have metaphase 1. That's when the homologous pairs line up. Sister chromatids don't line up, but homologous pairs, or the tetrads, line up. So the metaphase 1, and then anaphase 1, D, anaphase 1, this is when the homologous pairs separate. In mitosis, it's the sister chromatids separating, not the homologous pairs separating, so that's different. And then in prophase 1E, well, we've got genetic recombination, crossing over. That doesn't happen in mitosis. That only happens in meiosis. So the answer is C, D, and E, or just H. C, D, and E. All right. And then the last problem. In which steps of meiosis does the following occur? Chromosomes condense, mitotic spindle begins to form, nuclear envelope begins to break down, and crossing over occurs between recombinant sister chromatids. I highlighted this is important. Pause the video and figure out the answer. So the answer is, make the pen smaller a little. Where is it? Prophase 1. Prophase 1. That's when the crossing over occurs between recombinant sister chromatids. Alright, good luck. I hope this helped. Thank you for watching. Good luck.